Is the class rep still here? If you are here, please raise your hand so that I make you. It seems your network today has been disturbed. Okay, thank you. Okay, so neurohistology. Can we see the slide, please? Please, can we see the slide? I want to be sure if you can see it before I proceed. Can we see the slide? Class rep, can you see the slide? Can we see the slide?
Yes.
Okay, I'm coming to start.
Yes, sir, yes, sir. Yes, sir, we can see this. Yeah, we can see.
Okay. Now the nervous tissue or the nervous system is concerning any tissue that does some form of coordination in your body. For instance, you have been kissed and you found it so passionate and nice. It is the nervous tissue. You have been given a knock and the pain was so much. They are the nervous tissues. You are feeling body pains all over. Nervous tissues are telling you that. You are tired. Nervous tissues will tell your muscles that please relax. So what then makes this nervous tissue? Let us see how it is. So the very first thing now is when we take the nervous system, the nervous tissues are classified into two categories. We have the neurons and then the neuroglia. The neuron and then the neuroglia. The neurons are the main nervous tissues which connect or are responsible for the transduction of signals, while the neuroglia are the supporting cells for the nervous tissues. So neurons, they form an integrated communication of network, and they are responsible for receiving, processing, and transmitting sensory and motor informations. So neurons are actually the functional unit of the nervous system. They are the functional unit of the nervous system. So let us look at the structure, the neuron. So the neuron, when we take the neuron is made up of two main sides, a cell body and then an axon. So I'll quickly go to the whiteboard and try to see if I can demonstrate something. So when we take a neuron will look like this. It has a cell body. So this side is the pericardium, which is also known as the cell body. And then this is the axon of the cell body, of the neuron. This one is responsible for transfer of the information from the cell body to the muscle. So a muscle will be here. Then the cell body also has the receiving ends, which are known as the dendrites, and then So the smaller ones are the dendrites or the individual ones. The meeting point here is the dendron. So this is a dendrite. These are the dendrites. So you see the dendrites on the cell body or the tentacles. Then you have the cell body having a nucleus. You have the cell body having a nucleus in there. And then this nucleus will control the life of this cell. You have some rough endoplasmic reticuli and then mitochondria and thing all in the cell. But so let's go back to the slide. Class rep, if it is 11:40, you let me know so that I pause so that we have our 5-minute discussion. Okay. Today, class rep, your network is so bad. So it's okay. Just raise, alert me by raising your hand if the time is up. But where is the assistant? What's the name, Kra? Where is the assistant?
Please, I'm here.
Ah, Precious.
Yes, sir.
Okay. I can make you two a co-host.
Okay.
So I now have 2 co-hosts. Okay. So in case we can't hear from you, ma, I will call you. Okay.
Okay, sir.
Precious Corantin, is that not so?
Yes, please.
Thank you. Your network is a bit better. So let's continue. So let's go back to the slide. Oh, it seems okay. I've closed the slide. Let me quickly put it. Let's see. Okay. So this is what the man running on the football field or on his track. That if you remember that is how a neuron looks like. So you have the dendrites and then the legs over here. This is the axon which has been what? Divided.
So the screen can't be seen.
Oh,
So you can't see the screen.
Oh,
Yes, sir.
sorry. Please, can you see now?
Yes, sir.
Thank you very much. So I can carry on, eh?
Yes, sir.
Thank you. So we have the pericardium, which is the cell body or the soma. I've told you the things we can see inside already. So when we take a tissue like this, when you see something like this with all these things at the side, that is the nervous tissue or a neuron. Now those dark things that we are seeing here, they are known as the nuclear bodies. Okay, the nuclear bodies, and that is the nucleus. This is the nucleus itself, and this is the nucleus, and then the inside one is the nucleolus. And this whole thing here is the cell body. So you can only see the neuron in a connective tissue. So when you take any organ and you want to look for a neuron, you go to the connective tissue. That is where you see the neuron. That is where you see the blood vessels too. So that is that. So when we take the pericardium too, we have some of the regions, especially when we get to the brain regions, we have melanin secretory substances. Okay, and then we have something we also call the cytoskeleton of the neuron. They are actually made up of microtubules, just like the muscle, the same organization. So these microtubules allows the neuron to move. We will see a short view on that. And then the neurofilaments. So we have the neurofilaments, which are about 10 nanometers. They are also known as the intermediate filaments. And then we have the microfilaments. These are the cytoskeleton or the body of the neuron. So as we have myofibrils, we have neurofibrils. Neurofibrils are also clump of neurofilaments together. So this is just to show us the dendrites. This is the axon, and then the cell body, and then is the nucleolus, and this is the nucleus in general. So these are those dark things, nuclear bodies. The appearance of nuclear substance in a neuron indicates something. I want you to go and then look out for what it reveals when we talk about the nuclear bodies. Now the dendrites are very much branched, and then they are the receptive part for the neurons, and then they made lots of connection, a lot of synapses. Okay, and however, when you are aging and then you are diminishing, and you are aging, or you have poor nutrition, you have the spines of the dendrites becoming diminished. And when that happens, you begin not to feel senses at some certain parts of the body. Okay, so this happens to older people. Then the axon. The axon will actually project from the cell body. So axons can be very long, at times 100 centimeters in some larger animals. Okay, and then this part is what we call the axon hillock. Okay, you can see it's pyramidal in shape. Okay, so we call that part the axon hillock. And the hillock is made up of a lot of what? Regions that do not have the rough endoplasmic reticulum. But when you look at this, you can see a lot of ribosomes and then the rough endoplasmic reticulum. And when it comes to the hillock, you don't tend to see the rough endoplasmic reticulum. Now, let's look at the morphological categorization of neurons. Neurons can be classified based on 3 ways. We can classify neurons based on their morphology, that is their appearance, and we can also classify them based on their functions, and then their locations too. So based on their morphology, a neuron can be multipolar. That means it has more than one connection to the cell body. That's the most common type. Almost all neurons that carry information to the effector. When we say effector, that's the productor muscle organs, are multipolar in nature. Then bipolar, which we can find in the vestibular cortex and the cochlear ganglions in the brain. We will see that when we do the neuroanatomy. They are bipolar. Bipolar means they have 2 connections. You will see something like 2 axons. But the multipolar, we have a lot of mostly 1 axon and a lot of dendrites. So let's look at it over here. So this is a bipolar neuron. And then this is a multipolar neuron. This is one big multipolar neuron. You have call it multipolar because this part has a lot of dendrites and an axon too. But here you only have 1 and then 1 dendrite, 1 axon. Yeah, we call this one unipolar or pseudo-unipolar. This is actually pseudo-unipolar because it is giving us, if you look at it, you think it is bipolar. But actually, you only have 1 axon coming out from the cell body before it bifurcates. So this is pseudo-unipolar axon. Some neurons can also be named based on their shape. That's why we have pyramidal neurons, which can be found especially in the cortical center of the brain or in the hippocampus of the brain. And then we can find the Purkinje. This is how some of them look like, which can be found within the cerebellum of the brain. We'll look at this and then you'll see it better. So again, this is the motor neuron nerve ending. These are the dendrites. Okay, these are the dendrites. This is an axon. These are some form of neuroglia cells. So looking at a bit better one, whenever you go to the connective tissue and you have any round structure with a nuclei inside, and that round structure does not have a space, it's a neuron. It will appear reddish if it is H and E. If it is having a hole inside, then it is no longer a neuron, but it will be more of a blood vessel. So normally you have the neuron with a space around, okay. Okay, so let me speak on the synaptic junction. So when we say synaptic junction is made up of, let me go to the whiteboard. The synaptic junction is made up of 2 neurons coming to so this is the pre-neuron and then this is the post-neuron. And this is the synaptic cleft. So this is the presynaptic and then this is the postsynaptic axon. So this is the pre and then this is the post. This one will release its contents to communicate with us depending on what they are doing. Where it can be either adrenogenic or it can be cholinogenic. If you do begin your pharmacology, you will understand what I'm talking about. Adrenogenic means that they are going to release adrenaline. Cholinogenic, they are going to deal with acetylcholine. And then how their effects would be like. So that is the information on the presynaptic cleft. I'm going back to the slide. So we said in the synaptic junction, we have axon terminal from both sides. We have the presynaptic membrane containing a lot of mitochondria. They are going to release the thing. And then we also have the postsynaptic membrane, or they all contain the receptors that will receive the neurotransmitters from the presynaptic neuron. Okay. So this is just to show the inner part of it. So you can see this is the mitochondria. You can see the mitochondria in here. These are the microtubules. These are the microtubules. That's the building block. And then this is a vesicle which is going to release. These are the forming vesicles that are going to be released. They are going to release their substance, whether adrenaline or epinephrine or noradrenaline or norepinephrine or acetylcholine or mascarin or whatever. It depends on what they are going to release. And this is the postsynaptic cleft, which will have the receptors, which will pick them up and then transform them to whatever signal they want to transduct. When we talk about the neuroglia, I told you they are the supporting cells for the main neurons. So the neuroglia would have, we have 6 of them, 4 in the central nervous system and 2 in the peripheral nervous system. The 4 in the central nervous system are the astroglia, oligodendrocytes, microglia, and ependyma. All the Schwann cells and the satellite cells are in the peripheral nervous system. We will do these things in normal anatomy and you get to understand them, but you can read on them. You can only see this neuroglia by light microscopy, even without special staining. There are several glial cells to each neuron. That means that we have a 1 to several glial cells. When we have one neuron, we have a lot of what? Several glial cells. What could be their functions? Let's pick them one by one. The astrocytes, I told you they'll be in the CNS, precisely in the brain center, where their main activity is to maintain homeostasis, and then they produce glial scar at injury, and then they are also involved in the formation of the blood-brain barrier. The blood-brain barrier. So they're actually the largest we can find, the largest of all the nucleoglia, with small amount of heterochromatin. That means we don't have enough heterochromatin. We have a lot of intermediate filament also inside there. So what they do is that they provide structural and metabolic support. Structural and metabolic support to the neurons. And they act as scavengers of ions and neurotransmitters released into the extracellular space. Those that will begin to scavenge around and don't know what they are doing, they will take them up. So the astrocytes, you can see they are normally like this. We have protoplasmic astrocytes, which are normally associated with the blood vessels too. And then we have the perivascular or the fibrous astrocytes. Okay. Structural and metabolic support, you can see why we say that, because they are connected to the blood vessel. They will try to do metabolic activity for the neurons, which do not have that capacity to do that. So what we are saying here, these are astrocytes, the round ones there. Okay, these are astrocytes. You can see protoplasmic astrocytes in there. The next one is the oligodendrocytes. If you know the Schwann cells, whatever the Schwann cells do, that's what the oligodendrocytes will do, by in the central nervous system. So they are smaller than astrocytes, darker, with abundant rough endoplasmic reticulum, and then they will allow to enhance the electrical insulation in the central nervous system. So they will build up this, how do we call them? The Schwann cells, okay, for insulation. So our oligodendrocytes look more like this. And then the microglia. Microglia, they are normally involved in what? Pathologic activities, like whenever any foreign material comes in, they try to do justice. So looking at this, we are looking at our oligodendrocytes in there. We have our oligodendrocytes in there. This is the microglia. This is the microglia. So what I told you that they are APCs. When I say APCs, they are antigen presenting cells. So they are involved in what? Immunology of the brain, or immune production of the brain. Ependyma, they will line the holes, the cavities we found in the central nervous system, which we call the ventricles. And they are involved in what? The formation of what we call the choroid plexus. And that choroid plexus, which is also associated with the formation of what? Cerebrospinal fluid. Cerebral. So when you see something like this, a ventricle and all these things you see here are ependyma cells. Ependyma cells. Schwann cells, you know what they do? I will not talk much about it. They form the fatty substance that insulates the axon of a neuron to allow for insulation and enhancement. Interruption in what they form is what we call the myelin sheath. So the myelin sheath that they form, there is a break inside which we call node of Ranvier, and this is to allow for what? Faster movement of the nerve. So this brings us to the end of the nervous tissues, not the whole nervous tissues, but just the basic nervous tissues. We are now going to look at the main nervous, which is the brain, okay? So let me project that one.